This video demonstrates the movement of sister chromosomes and homologous pairs during mitosis. There's a similar video which diagrams sister chromatids and homologous pairs in meiosis and which compares mitosis versus meiosis. In our cell here, we start out with two chromosomes. We have chromosome 1, which is here, and then chromosome 2, which is here, and chromosome 2 is much shorter. We have one copy of each chromosome from each parent. So the chromosome that is blue is from the mother, and the chromosome which is red is the father. Now, this is the situation when the cell is in G1 phase, for example, before S phase, where we have two autosomes, and again, that means we have two chromosomes of different lengths, and then we have two copies of each one from each parent. These two copies are called homologs or homologous chromosomes. Now, we've already gone over this in class, but I just want to remind all of us. So these can be called homologous chromosomes or homologous pairs, either one. And so this would be another homologous pair. And so in total, this cell has two homologous pairs. And that will never change no matter what stage of the cell cycle that we're talking about. After S phase, we have copied the DNA, and we're going to have duplicated all of the pieces of DNA that we had previously. So we have another copy of this, we have another copy of this, and then we have a second copy of this, and a second copy of this. So now we have two chromosomes or two copies of chromosome 1 from mom, and these will be connected during mitosis. And we have two copies of chromosome 2 from mom, and then we have two copies of chromosome 1 from dad, and two copies of chromosome 2 from dad. So if we were to say that we wanted to look at a specific gene, perhaps chromosome 1 has genes for height, maybe the eye color, and whether you taste something soapy when you taste cilantro or if you just taste like a spice. Okay, so for all these characteristics, for these three characteristics, there are multiple what we call alleles or multiple variations. And we'll talk about that in a couple of classes. Just because chromosome 1 from mom has the gene for height there, and chromosome 2 from dad also has the gene for height, it doesn't mean it's the exact same DNA sequence. From mom, we might get a short gene. I know that I got a short gene from my mom. And from dad, you might get a tall gene. So you can have different DNA sequences, but in that same place on chromosome 1, you're going to have a gene for height. So let's also look at eye color. And so we'll say that the eye color is right at the end of the chromosome, of chromosome 1. And so again, maybe from mom you get a gene for brown eyes, and then from dad you also get a gene for brown eyes. And so again, the gene is in the same position on the chromosome, but it doesn't mean that the DNA has to be identical. In addition, or rather in juxtaposition, it's different for the sister chromatids, or rather it is the same for the sister chromatids. What do I mean by that? Well, this sister chromatid is from mom, and this one is also from mom, and they're both for chromosome 1 or autosome 1. So they will have the exact same DNA sequence for height here as well as for eye color. The chromatid from dad and the other chromatid or sister chromatid from dad are going to have identical DNA sequences. And so I'm just going to write ID, and then similarly, I'm going to write ID here for mom to signify that the chromatids are the same. And again, you could call them sister chromatids. Now, what's going on here with chromosome 2? Well, chromosome 2 is an entirely different chromosome. So maybe that's where we find the gene for a soapy versus a spicy cilantro taste, or I guess mild cilantro taste. So maybe that is here on chromosome 2. So maybe mom's giving you a gene where you taste the soapiness, and then dad is giving you a gene where you also taste the soapiness. So you're getting the same gene sequence most likely from both parents, but in other places on those chromosomes, they might differ. And also, again, notice this is why for the homework assignment, I told you to make the autosomes different lengths just to illustrate that they have different gene sequences. Because on chromosome 1, we have this gene for height and eye color for no matter which parent you get it from. But then on chromosome 2, you have completely different genes. You have different traits, and we theorized here or imagined here that we have the gene for tasting cilantro. Okay, so this is after S phase. We have 4 copies of each gene, but not necessarily 4 identical copies of DNA, because we have 2 copies of hair color from mom, 2 copies of hair color from dad. These 2 might be identical. These 2 might also be identical to one another, but these 4 don't have to be identical. And then similar with chromosome 2. Chromosome 2 chromatids might be similar and have the same genes, but only sister chromatids, those from the same parent, are going to be identical. So in mitosis, after metaphase, we would get the separation of sister chromatids. So we would just erase this connection. In meiosis, we do something different.